Pod Microbiology Practical 3. Determining numbers of microorganisms in a sample. Method 1. Pour plate method. The objective of this experiment are: first, to learn how to determine the number of microorganisms in a sample, a process called enumeration. Secondly, to utilize several methods for enumerating microorganisms. The introduction of pour plate method, which is pour plate method overview. Technique for isolating and counting the viable microorganism present in a liquid sample, which is added along with or before molten agar medium prior to its solidification. Then, it is a simple method, less resource consuming, easy and economical. The procedure of the pour plate method is like this. A sample of non-dilution is mixed with a liquefied agar medium. Then, a liquefied agar medium is poured into sterile petri plate. The isolation and enumeration in pour plate method allows for the isolation of the viable colonies, and after the incubation, the colonies develop and can be counted to determine the concentration of the bacteria in a sample. These are the materials used in pour plate method for this experiment. Label 3 of the 99 ml dilution blanks, turn -2, turn -4 and turn -6 respectively. Label the 4 petri plates turn -2 through turn -5. Aseptically remove 1 ml of sample with a sterile pipette and transfer to the turn -2 dilution blank. Shake the dilution blank vigorously. Using a new sterile pipette, aseptically transfer 0.1 ml and 1 ml from the turn -2 dilution to the agar plate labeled turn -3. Turn -3 is 0.1 ml and turn -2 is 1 ml respectively. With the same pipette, remove an additional 1 ml from the turn -2 dilution blank and transfer to the turn -4 dilution blank. Repeat the shaking procedure for the turn -4 blank and transfer 0.1 ml and 1 ml portion from this dilution bottle with a new pipette to the plates labeled turn -5 0.1 ml and turn -4 1 ml. After that, aseptically pour a tube of melted agar into each petri plate to which we have already added a dilution of the sample. Swirl the plate to mix the sample with the agar and then incubate the inverted plate at 30 degrees Celsius. After incubation, count the colonies on each plate. At the end of the experiment, after the incubation, the number of bacteria in a sample was counted. And this is the result of our experiment for pour plate method. The number of count 1 and count 2 and average was all the same for all the dilution. Dilution of 10 to the power of -2 have 21 bacteria. Dilution 10 to the power of -3 have 5 bacteria. Dilution of 10 to the power of -4 have 133 bacteria, while the last is dilution of 10 to the power of -5 have 39 bacteria counted. Thus, based on the result, dilution of 10 to the power of -4 have the highest number of bacteria, while dilution of 10 to the power of -3 have the lowest number of bacteria. To calculate the concentration of bacteria in the original sample, the number of bacteria counted in the dilution times the number of dilution. For example, for dilution 10 to the power of negative 2, 21, which is the number of bacteria counted, times 100. 100 is same as 10 to the power negative 2, which is the dilution. So equal to 2100 bacteria per ml. Same as dilution, the other dilution. For dilution of 10 to the power of negative 3, have 5,000 bacteria per ml. For dilution of 10 to the power of negative 4, have 1,333,000 bacteria per ml. For dilution of 10 to the power of negative 5, have 3,900,000 bacteria per ml. So for the concentration in a sample, for concentration of bacteria in the original sample, we calculate the mean of all these for dilution and get 1,309,275 bacteria per ml for this 4-plate method experiment. This is the picture of our result in 4-plate method experiment. The discussion of this experiment is the number of microorganisms is calculated by enumeration from the sample of population. In the 4-plate method, a petri dish containing 1 ml or 0.1 ml of diluted bacterial suspension is added as a representative sample of the population. 4-plate method cannot be used only in enumeration since limitations of 4-plate methods will damaging or killing heat-sensitive bacteria. Consequently, no discernible colonies were discovered. Aerobes cannot survive because they are trapped inside the agar, preventing oxygen from penetrating or diffusing into it. Based on our result, the concentration of bacteria in dilution 10 to the power of negative 2 is 2,100 bacteria per ml, 5,000 bacteria per ml in dilution of 10 to the power of negative 3, 1,330,000 for dilution of 10 to the power of negative 4, and 3,900,000 bacteria per ml in dilution of 10 to the power of negative 5. The mean for the concentration of bacteria in the original sample is 1,309,275 bacteria per ml. Thus, we can conclude that the dilution of 10 to the power of negative 4 have the most bacteria growth, which in our result 133 CFU compared to the dilution of 10 to the power of negative 3, which have the least bacteria growth. The bacteria also in the dilution of 10 to the power of negative 4 seem more crowded than the other dilution, which is dilution 10 to the power of negative 2, 10 to the power of negative 3, and 10 to the power of negative 5, which the less crowded bacteria from dilution of 10 to the power of negative 4. Lastly, the agar is left to be cooled and still molten before poured into the bacterial suspension to prevent the damaging of bacteria. If the agar is too hot, the bacteria in the sample may be killed. If the agar is too cool, the medium may be lumpy once solidified. For 4-plate methods, first question, why are counts above 300 and below 30 statistically unreliable? The answer is, in statistics, the sample size is inversely proportional to the margin of error. Thus, below 30 colonies per plate has a larger error in the results. If there are too many colonies in an agar plate, the closer colonies may form a single colony, or only one colony will grow as competing for nutrient with each other. Thus, above 300 colonies per plate is statistically unreliable too. The second question is, did you enumerate all the bacteria using this method? The answer is no. The poolplate method is commonly used for the microbiological control of food, cosmetics and pharmaceutical products, and particularly for the enumeration of microorganisms with low microbiological limits. For example, Staphylococcus aureus in food samples or mesophilic aerobic bacteria in drinking water. Enumerate heat-sensitive bacteria using this method is not suitable as it may kill them. Question is, what factors make the poolplate method selective? How can you reduce the selectivity of this procedure? The answer is, poolplate method might kill the heat-sensitive microbes with hot agar. To reduce the selectivity of this procedure, the agar is left to be cool but still molten before poured into the petri dish. The fourth question is, how can you achieve a 1 per 100 dilution other than by adding 1 ml of sample to a 99 ml dilution blank? The answer is, add 0.1 ml of sample to the 9 ml dilution blank, or add 1 ml of sample from the 1 per 10 dilution to a 9 ml dilution blank.
Practical 3. Determining numbers of microorganisms in a sample by using spread plate method. One common method used in microbiology to ascertain the number of bacteria in sample is the spread plate method. When evaluating the microbial load of materials such as food, water, or clinical specimen, this technique is especially helpful. Using a sterilized glass rod or spreader, the method entails uniformly spreading a known volume of the sample across the surface of a solid agar medium. The objective is to disperse germ uniformly so that viable colonies may be isolated and counted. Following incubation, colonies grow and become countable, allowing for an estimation of the original sample's microbial population. These are the materials used in this experiment. Procedure. Label 3 of the 9 ml dilution water blank as 10-1 to 10-3 respectively, and label 4 nutrient agar plate from 10-1 to 10-4. Label 3 of the 9 ml dilution water blank as 10-1, 10-2, and 10-3 respectively, and then label 4 nutrient agar plate from 10-1 to 10-4. After that, aseptically pipette 1 ml of the water sample into the 10-1 dilution tube, and then mix the 10-1 dilution tube thoroughly by vorticing or vigorous shaking. Using the same procedure, transfer 1 ml from the 10-2 dilution tube to the 10-3 dilution. Protect the sample then. After that, using a new sterilized pipette, transfer 0.1 ml from each of the dilution tube and then original sample to the agar plate. After that, sterilize the glass spreader by dipping it in alcohol and flaming it in a Bunsen burner flame. After the glass rod has cooled, spread the sample over the surface of the plate by touching the rod to the agar and rotating the plate. This procedure is repeating for each of the plate. After finish with the procedure, incubate the plate at inverted at 30 degrees Celsius for 24 to 48 hours. After incubation, count the colonies and then calculate the concentration of bacteria in the original sample. Result. For the dilution of 10-1 is too numerous to count as the average, and the second one is dilution of 10-2 having 244 colony forming unit, and 10-3 is 190 for the colony forming unit, and the last one is 10-4 having 172. To calculate the concentration of bacteria in the original sample is by multiplying the colony forming unit with the dilution factor. The concentration of the bacteria in the original sample for each dilution is calculated. Then add all the concentration of the bacteria in the each dilution. Then we divide it by 3 to get the mean of the concentration of the bacteria in the original sample, which is we get 644,800 bacteria per mil. And this is the result of the spread plate method. Next is discussion part. The spread plate method calculated mean bacteria concentration of 644,800 bacteria per mil collected across 3 dilutions highlight the method's effectiveness in delivering a quantitative evaluation of microbial population. The highest dilution will show less microbial growth compared to the lowest dilution, where there are high concentration of bacteria in the sample. For example, in the dilution of the 10 negative 1 from the experiment, there are two numbers to count for the dilution 10 to the power negative 1, while for the dilution of 10 to the power of negative 4, there are only 172 colony forming unit can be calculated. The answer for first question is colony forming unit represent both single cells and cluster capable of forming a colony, providing a more accurate measure of valuable cells in sample, while colony is a visible cluster of microorganisms in the spread plate method. An answer for the second question is cross-contamination error. It can happen when using the same pipette for different samples. The reliability of the result could be affected if excess material from one sample continue into the next. Question 3. The advantages of the spread plate method is first one is the ease and simplicity, second one is isolation and enumeration, and the third one is applicable to bacteria and fungi. While for the limitation of the spread plate method is it is only limited to surface growth and the incubation time consume 24 hours to 48 hours. For the last question is how can you modify the spread plate method procedure to enumerate the obligate anaerobe is by using specialized anaerobe culture medium such as reinforced clostridial agar, or we can use a sterilized method. In summary, the goal of understanding the technique of enumeration to determine the quantity of microorganisms present in a sample has been accomplished. A solid understanding of microbial quantification has been established by means of the investigation and application of diverse enumeration techniques, including the spread plate method and pour plate method. Application in many fields such as microbiological research, quality control procedures, and guaranteeing the safety of goods like food and water require this understanding. Finally, from this experiment, we can determine the heat-sensitive microbe by pour plate, while non-heat sensitive microbe by spread plate method.